Okay, the next topic we're going to talk about are proteins. This is the third class of macromolecules. So there's a video that was made for carbohydrates and the introduction into macromolecules. The second video that was made was lipids. Now we're going to take a look at proteins. Unlike lipids, lipids was the off one. That was the one that was kind of different. Carbohydrates have a monomer and proteins have a monomer. That means they have a building block, something that they're all made up of. The monomer for a protein is an amino acid. So there are actually 20 different amino acids. And of those 20 different amino acids, they form all different kinds of compounds when they come together, such as antibodies, which you've probably heard of, hormones, enzymes, and even parts of muscle that we're going to take a look at later. So again, they've got so many different functions because there's so many different proteins, I can't even go into all the different functions or all the different types of proteins. It'd be similar to saying, okay, you've got about 26 letters of the alphabet, and look at how big the dictionary is and how many words there are. So same kind of idea. You can put these 20 different amino acids in different orders. You can have one protein that might have 1,000 amino acids and another protein that has like 1,001, and that makes a whole different protein. So just the number of amino acids and the order of amino acids makes a different protein. And keep in mind, these are built by the body. Ribosomes make proteins. Ribosomes are a component found in cells, and they actually make the proteins. So how do they know how to make them? This is the direction of the nucleus. So the nucleus has the instructions on how to make all these proteins, how to put the amino acids in the right order and the right number, and then the ribosome does the work. So this is stuff that the body makes. This is the general structure of an amino acid. So all 20 amino acids have this same general structure. It's got an amino group on one end. And how do you know it's an amino group? Nitrogen. That nitrogen is present. So that tells you, oh, that's an amino group. It's got a carboxylic acid group on the other end. So you're looking at this carbon right here, this alpha carbon. And attached to that carbon, you've got four things. One is the amino acid group. The other is the carboxylic acid group. So you can kind of see where the amino acid gets its name from. Amino on one side, acid on the other. That's where the name of the compound comes from. And then off of another side of this alpha carbon, you've got a hydrogen. So those three things are the same. They all have the amino group, they all have a hydrogen, and they all have that same carboxylic acid. What's different about them is that fourth thing coming off that alpha carbon, which is the R chain. So each of the 20 different amino acids, what makes them different is that R group. It could be another just plain hydrogen. It could be a whole hydrophobic fat loving compound. It could be a water loving compound. Could be an acid coming off that carbon. Could be a base coming off that carbon. This was just one of the funny things. This is still got what you have here is that carbon right there, that alpha carbon with all the different things coming off of it. You've got the amino group, you've got the carboxylic acid, and then the invisible hydrogen that you can't see. And then this right here is that R group. We do need to mention enzymes. We've talked about enzymes before and about how enzymes are very picky molecules, meaning they really like to be at that 98.6 temperature in the body. And if they get somewhere outside that range, they're not going to function anymore. So enzymes are a special type of protein. Again, they're still made by amino acids linked together, but these are easily denatured depending on the conditions. Now, how do you know when you've got an enzyme name? ACE, ASE. Anytime you have a word that ends in ASE, you know that you have an enzyme. Now, and when we're talking about proteins, again, going back to the main thing, not just the special type of protein called an enzyme, but overall proteins, there are 4 levels of protein structure. You have primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure, you need to know that it is just literally the sequence of amino acids. So it's literally just telling you what amino acid comes next in the sequence. That's it. With secondary protein structure, this is formed from hydrogen bonding. So this has to do with the um, pleated sheets and the alpha helix. So usually they're called beta pleated sheets and alpha helices. So it's the turning and the folding that occurs from the protein, and that has to do with the interactions of the hydrogen of one thing bonded to the oxygen, fluorine, or nitrogen of something else, and that just forms these different kinks and twists in the protein. And then tertiary structure, this is due to all types of interactions, and you're going to get the actual functional protein due to the folding at the tertiary level. And then quaternary structure is when you have 2 or more proteins that are bonded together. So you can see in the picture here, you've got protein number 1, which would be a light colored protein, and then protein number 2, which would be a different kind of the dark blue protein, and they've come together and formed a larger structure. So that's quaternary structure. So again, this is just the same, different view, same thing on this slide here. That's it for proteins.